Okay. Hello guys, I'll share my screen and one of you can unmute and let me know if I'm audible and if my screen is visible. Okay. So can one of you please let me know if my screen is visible and if I'm audible? Okay. So good evening everyone. Today is the third live session of this course, RNA Biology. So as we know, in these live sessions, we discuss some previous year assignment questions. We discuss not only the answers but also the concept behind those answers so that it's easier for us to solve similar type of other questions, right? So that's what we'll be doing in this session as well. So we'll start with the session. We want to keep the sessions as interactive as possible. So if you have any questions, you can just unmute and let me know. Raise your hand, or write in the chat box. I'll get back to you. Right. So let's start today's question. So this is the first question for today. RNA-sp is important for RNA stabilization, free tRNA maturation, ribozyme action, or nitrogen base modification. So only one answer is correct. I'll give you 2-3 minutes to look into the question and tell me the answer. Then we'll discuss the concept behind those questions. Behind the question. Okay, so the answer will be P pre-tRNA maturation. So ribonuclease P is basically ribozyme that cleaves RNA to process transfer RNA, that is tRNA. It's a ribonucleoprotein that contains both RNA and protein subunits, right? So it cleaves the 5' leader sequence from pre-tRNA. It processes the 5' ends of precursor tRNAs and also helps in the maturation of the 5' end of tRNA. It's found in cells from all three domains of life: bacteria, archaea, eukarya, etc. And so how it works, we'll discuss it in a few minutes. So we'll talk about the biography of ribonuclease P. So the discovery was As you can see the photo of the gentleman over here, Sidney Altman, and you can go through this paper. Altman published in 19 Altman et al. Published in 1971. Okay, so this is, you can see RNA-SP, RNase actions, purifying RNA-SP, actions of purified RNA-SP. So how it works? RNA subunit of RNA-SP is catalytic and acts like a protein-based enzyme. So the RNA subunit folds into a three-dimensional structure that forms a cleft to bind the substrate. Now the specificity domain of the RNA subunit recognizes the substrate. The catalytic domain of the RNA subunit contains the active site. Okay, so you can see Benjamin Stark, who was a graduate student, identified the RNA and protein subunit of E. coli RNA-SP. You can see M2 stands for methylene blue staining for nucleic acid stain, and C5 is coumacy brilliant blue staining for protein. Okay. So RNA, as I said, ribonuclease P is a type of ribonuclease which cleaves RNA. RNase P is unique from other RNases in that it is a ribozyme, a ribonucleic acid that acts as a catalyst in the same way that a protein-based enzyme would. Its function is to cleave off an extra or precursor sequence of RNA on tRNA molecules. Further, RNase-P is one of the two known multiple turnover ribozymes in nature, the other being the ribosome, the discovery of which earned Sidney Altman and Thomas Craig the Nobel Prize in Chemistry in 1989. In the 1970s, Altman discovered the existence of precursor tRNA with flanking sequences and was the first to characterize RNase-P and its activity in processing of the 5' leader sequence of precursor tRNA. Recent findings also reveal that RNase-P has a new function. So it has been shown that human nuclear RNase-P is required for the normal and efficient transcription of various small non-coding RNAs, such as tRNA, 5S rRNA, SRP RNA, and U6SN RNA genes, which are translated by RNA polymerase III, one of the three major nuclear RNA polymerases in human cells. Right. So bacterial RNase-P has two components, an RNA chain called M1 RNA and a polypeptide chain or protein called C5 protein. In vivo, both components are necessary for the ribozyme to function properly, right? But in vitro, the M1 RNA can act alone as a catalyst. The primary role of the C5 protein is to enhance the substrate binding affinity and the catalytic rate of the M1 RNA enzyme, probably by increasing the metal ion affinity in the active site. The crystal structure of a bacterial RNase-P holoenzyme with tRNA has been recently resolved, showing how the large coaxially stacked helical domains of the RNase-P RNA engage in shape-selective recognition of pre-tRNA target. This crystal structure confirms earlier models of substrate recognition and catalysis, identifies the location of active site, and shows how the protein component increases RNase-P functionality. Bacterial RNase P class A and B. So ribonuclease P is a ubiquitous endoribonuclease found in archaea, bacteria, and eukarya as well as chloroplasts in mitochondria. Its best characterized activity is the generation of mature 5' ends of tRNAs by cleaving the 5' leader elements of precursor tRNAs. Cellular RNase-P are ribonuclear proteins. RNA from bacterial RNase-P retains its catalytic activity in the absence of a protein subunit, that is, it is a ribozyme, right? So isolated eukaryotic and archaeal RNA-P RNA has not been shown to retain its catalytic function, but it's still essential for the catalytic activity of the holoenzyme. Now, although the archaeal and eukaryotic holoenzymes have a much greater protein content than the bacterial ones, RNA codes from all the three lineages are homologous. Helices corresponding to P1, P2, P3, P4 and P10, P11 are common to all cellular RNAs. pRNAs. Here there is a considerable sequence variation, particularly among the eukaryotic RNAs. Now in RNA, RNA-sp ribonuclear proteins consist of 4 to 5 protein subunits that are associated with the RNA. As revealed by in vitro reconstitution experiments, these protein subunits are individually dispensable for tRNA processing, that is essentially mediated by the RNA component. Structures of protein subunits of archaeal RNA-sp have been resolved by X-ray crystallography and NMR, thus revealing new protein domains and folding fundamental for function. Using comparative genomics and improved computational methods, a radically minimized form of the RNA-sp RNA, dubbed type T, has been found in all complete genomes in the crenarchaeal phylogenetic family Thermoproteaceae, including species in the genera Pyrobaculum, Cultivirga, and Vulcaniceta. All retain a conventional catalytic domain but lack a recognizable specificity domain. 5' tRNA processing activity of the RNA alone was experimentally confirmed. So the Pyrobaculum and Cultivirga RNAs are the smallest naturally occurring form yet discovered to function as trans-acting ribozymes. So loss of the specificity domain in these RNAs suggests potential altered substrate specificity. It has recently been argued that the Archaebacterium nanoarchaeum equitans does not possess RNA-sp. Computational and experimental studies fail to find evidence for its existence. In this organism, the tRNA promoter is close to the tRNA gene, and it is thought that transcription starts at the first base of the tRNA, thus removing the requirement for RNA-sp. Next, in eukaryotes such as humans and yeast, most RNA-sp consists of an RNA chain that is structurally similar to that found in bacteria, as well as 9 to 10 associated proteins. So 5 of these protein subunits exhibit homology to archaeal counterparts. So these protein subunits of RNA-sp are shared with RNase MRP, a catalytic ribonuclear protein involved in processing of ribosomal RNA in the nucleus. So RNA-sp from eukaryotes was recently demonstrated to be a ribozyme. Accordingly, the numerous protein subunits of eukaryotic RNA-sp have a minor contribution to tRNA processing per se, while they seem to be essential for the function of RNA-sp and RNase MRP in other biological settings such as gene transcription and cell cycle. Despite the bacterial origins of mitochondria and chloroplasts, plastids from higher animals and plants do not appear to contain an RNA-based RNA-sp. It has been shown that human mitochondrial RNA-sp is a protein and does not contain RNA. Spinach chloroplast RNA-sp has also been shown to function without an RNA subunit. Okay. So you can see RNA-sp functions to remove extraneous 5' sequences from precursor tRNAs to generate the mature tRNA. You can go through this paper as well, Evans et al. 2006. Now the mechanism of function is pre-tRNA binding to S and C domains of RNA-sp RNA subunit, RNA-sp protein subunit binding to 5' end of pre-tRNA. Now what is the function? A human RNA-sp transcription factor. RNA-sp protein found at 5S RNA, 7SL RNA and U6 SN RNAs, non-RNA-sp substrate genes and tRNA genes, RNA-sp substrate genes. So what is the potential as a tool? Potential as an antibacterial, antiviral and anti-cancer agent. Now can we take advantage of the catalytic function of M1 RNA to target bacterial viral oncogenic mRNAs? So we'll talk about guide sequences. We have the external guide sequences, exogenous GS, recruits endogenous RNA-sp. We have internal guide sequences, GS covalent linked to M1 RNA. Now, what are the requirements? Complementary to target mRNA, 3' sequence for recognition by M1 RNA. If you talk about application, antibacterial potential, Carrier Takada et al. 1995 shows specific targeting of EGS, 2-beta-galactosidase, and alkaline phosphatase encoding genes. Expressions were decreased by 50-60% in E. coli. If you talk about the antiviral potential, IGS and EGS, internal guide sequence and external guide sequences, were used to target herpes simplex virus HSV1, human immunodeficiency virus HIV, human influenza virus, human cytomegalovirus, and Kaposi sarcoma associated herpes virus. Anti-cancer potential M1GS used for destruction of chimeric mRNAs created by chromosomal translocation BCR-ABL. So there is a lot of anti-cancer application as well. RNA-sp is now being studied as a potential therapy for diseases such as herpes simplex virus, cytomegalovirus, influenza, and other respiratory functions. HIV and cancer caused by fusion genes. So the external guide sequences are formed with complementarity to viral or oncogenic mRNA and structures that mimic the T-loop and accept the stem of tRNA. So these structures allow RNA-sp to recognize the EGS and cleave the target mRNA. EGS therapies have shown to be effective in culture and in live mice. So if we talk about application advantages for basic science research, so as I said, it's highly specific and does not mistarget RNA targeting concept. The little sense of cytotoxicity and external guide sequence also uses endogenous RNA-sp, most abundant, stable and efficient enzymes resulting in irreversible cleavage of target mRNA. Okay, so coming back to the question at hand, RNA-sp is important for pre-tRNA maturation. Okay, if anybody has any doubt, you can ask me now. I'll wait for 2-3 minutes, then we'll move on to the next question for today. Okay, so there's no question for this. We'll move on to the next question for today. During protein synthesis in ribosomes, peptide bond formation is by protein, tRNA, RNA, or mRNA. Which one do you think is the correct answer? I'll again wait for 2-3 minutes. If you know the answer, please let me know. Sorry for my sore throat. So if you know the answer, you can just unmute and let me know. Or we'll start in 2-3 minutes. Anybody? Okay, so the answer will be obviously RNA. So, proteins are polymers made up of amino acids linked together by peptide bonds, and these are the most important organic substances in a living cell. They have a high molecular weight ranging up to several millions. So, proteins perform various functions such as enzymatic function, hormonal function, etc. Okay, so this is the structure. If you see, this is the peptide bond. CONH, this is the carboxyl terminus, C terminus, amino terminus, and N terminus. This is the backbone, these are the side chains. Now, amino acids are the building blocks of proteins. They have one amino group and one carboxyl group. Other functional groups may also be present. There are about 20 amino acids which make all proteins on Earth, and each amino acid has 4 different groups attached to a carbon. These 4 groups are hydrogen, amino group, carboxyl group, and a side chain. Hydrogen, amino group, carboxyl group, and a side chain, right? So this R represents functional group. Example, in glycine it's H, serine it's OH, and in alanine it's CH. So there are various different types of amino acids, polar, non-polar. So 20 amino acids are commonly found in protein. These 20 amino acids are linked together through peptide bond forming peptides and proteins. So the chains containing less than 30 amino acids are called peptides, while those containing greater than 30 amino acids are called proteins. Now, how does the peptide bond formation occur? So carboxyl group of one amino acid forms a covalent peptide bond with amino group of another amino acid by removal of a molecule of water. The result is dipeptide, that is two amino acids linked by one peptide bond. So in the same way, the dipeptide can then form a second peptide bond with the third amino acid called tripeptide to give a tripeptide basically. So repetition of this process generates a polypeptide or protein of specific amino acid sequence. So this is how the peptide bond formation will occur. Now the structure of protein, what are the levels of protein structure? So there are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. So the primary structure is a linear sequence of amino acids forming a polypeptide chain. At one end is an amino acid with a free amino group, the N terminus, and at the other end is an amino acid with a free carboxyl group, the C terminal. So only a few plant proteins occur in this form. Now, a functional protein is not just a polypeptide chain, but one or more polypeptides precisely twisted, folded, and coiled into a molecule of unique shape. So, this conformation is essential for some protein function. Now, if we talk about secondary structure, most long polypeptide chains are folded or coiled. So, folded or coiled. So, this brings about the secondary structure. The folding of the polypeptide chain is maintained by hydrogen bonds formed between adjacent amino acids. Alpha helix, it is the most common form of coiling. It is right-handed coil strand. It is stabilized by hydrogen bonds between H of NH2 group and O of CO group of 4th amino acid away in the peptide shape. So although hydrogen bond is weak, the large number of maintains a stable structure. That is, example, keratin in hair, myosin in muscles, etc. Now, if we talk about beta pleated sheet, it is another form of secondary structure. It is a sheet-like structure. It's not rod-like. So the hydrogen bonding is in between strands, interstrand, rather than within strands, like intrastrand. So pairs of strands lie side by side. So the hydrogen bonds occur between H of NH2 group of one strand and oxygen of carboxyl group of another strand. The two strands may be parallel or antiparallel. For example, silk fibroin. Now, if we talk about tertiary structure, example myoglobin, it involves folding and coiling of polypeptide chain to produce a complex globular shape. So this structure is maintained by 4 types of bonds. Disulfide bonds, SS bond, is formed between two cysteine residues. Hydrogen bonds formed between the H of NH2 group and O of CO group. Ionic bonds formed when an acidic and a basic amino acid are ionized and lie together. Hydrophobic bonds formed because of tendency of nonpolar side chains of neutral amino acids to closely associate with one another. hydrophobic interactions, clustering of hydrophobic groups away from water, and van der Waals interactions. Now, if you talk about quaternary structure, it refers to the association of two or more polypeptide chains to form a stable unit. Now, if you talk about identical units, homogeneous proteins, example isozymes H4 and M4 of lactic dehydrogenase. Dissimilar units, heterogeneous proteins, example hemoglobin, 2 alpha chains and 2 beta chains. All of that. Now, if you talk about the biological functions of proteins, there are membrane proteins. Proteins and lipids form the major components of cell membranes. Many enzymes are associated with the membranes. Enzymes, all the enzymes are proteins, they are biocatalysts. Hormones, many hormones and peptides are proteins. They play an important role in regulation of metabolic activities. Blood proteins include the plasma proteins and hemoglobin. Antibiotics like gramicidin S, thyrocidin, and penicillin G are peptides. And nucleoproteins, these are conjugated proteins, proteins plus nucleic acids of a cell nuclei. So if you talk about 20 amino acids commonly found in proteins are joined together by peptide bonds. The linear sequence of the linked amino acids contains the information necessary to generate a protein molecule with a unique three-dimensional shape. So the complexity of protein structure in four organizational levels, primary, secondary, tertiary, and quaternary, as shown in this figure. These are the levels of protein organization. Primary protein structure is a sequence of chain of amino acids. Secondary protein structure occurs when the sequence of amino acids are linked by hydrogen bonds. Tertiary protein structure occurs when certain attractions are present between alpha helices and beta pleated sheets. And quaternary protein structure is a protein consisting of more than one amino acid chain. Now, if we talk about the primary structure, it's defined as a sequence of amino acid proteins. Now, understanding the primary structure of protein is important because many genetic diseases result in proteins with abnormal amino acid sequence, which will cause error, folding, and loss or impairment of normal function, right? So, in proteins, amino acids are joined covalently by peptide bonds, which are amide linkages between the alpha carboxyl group of one amino acid and the alpha amino group of another. So, for example, the formation of the peptide bond between glycine and alanine. Okay. Peptide bonds are broken by prolonged exposure to strong acid and base at high temperature to hydrolysis. These bonds non-enzymatically. Okay, so this is a peptide amide synthesis. Now, naming the peptide bond, free amino end is written on the left and free carboxyl end is written on to the right. Therefore, all amino acid sequences are read from the end to C terminal end of the peptide. Now, polypeptide linkage of many amino acids through peptide bonds results in an unbranched chain. So, residue or moiety, each component amino acid in a polypeptide. So, when a polypeptide is named, all residues have their INE, ANIC or ATE changed to YL with exception of the C terminal amino acid. So, what are the characteristics of the peptide bond? Partial double bond character, that is, it's shorter than single bond. Rigid and planar, this prevents free rotation around the bond between carbonyl and nitrogen of peptide bond. Trans configuration because of the steric interference of the R groups when it is in cis position. Uncharged but polar. Now, determination of the amino acids composition of polypeptides. So, the determining of primary structure of polypeptide is to identify and quantitate its constituent amino acids. So, by hydrolysis with a strong acid at high temperature for 24 hours, to release the individual amino acids which can be separated by cation exchange chromatography. A mixture of amino acids bind to the column that contain a resin which are negatively charged group with different affinities depending on their charges and hydrophobicity. So separated amino acids contained in the yellowette with solutions of increasing ionic strength and pH are quantitated by heating them with nitrogen, a reagent that forms a purple compound with most amino acids. The amount of amino acids is determined spectrophotometrically by measuring the amount of light absorbed by the anhydride derivatives. So the analysis described is performed using an amino acid analyzer as shown in this figure. You see over here. Now, how does a sequencing of the peptide form from its N-terminal end? So, beginning at the N-terminal end and phenyl isothiocyanate, known as the Edmonds reagent, so Edmonds degradation happens. So, it's a method of sequencing amino acids in a peptide. In this method, the amino terminal residue is labeled and cleaved from the peptide without disrupting the peptide bonds between other amino acid residues. So, the method can be employed by a machine called sequenator. This is PITC or phenyl isothiocyanate polypeptide, and then this is PTC polypeptide, and this is how it's schematic diagram of the Edmond degradation. So change of the polypeptide into smaller fragments. Many polypeptides are structure composed of more than 100 amino acids. These are large molecules can be cleaved at specific sites and the resulting fragment sequenced by using more than one cleaving agent, enzymes or chemicals. Now, determination of protein's primary structure by DNA sequence. The nucleotide sequence can be determined by knowledge of genetic code and translate into corresponding amino acid sequence of the polypeptide. So this method has limitations of not being able to predict the positions of disulfide bonds in the folded chain and not identifying any amino acids that are modified after their incorporation into the polypeptide. Is defined as regular arrangement of amino acids that are located near to each other in the linear sequence. Secondary structure, right? The alpha helix, the beta sheet, and the beta bend are examples of secondary structure. So in case of alpha helix, is a spiral structure consisting of a tightly packed coiled polypeptide backbone core with the side chains of the component amino acids extending outward from the central axis to avoid interfering sterically with each other. So the telephone cord shape of the alpha helix is held in place by hydrogen bonds between every NH group and the oxygen of a CO group in the next turn of the helix, 4 amino acids down the chain. The typical alpha helix is about 11 amino acids long. Okay. So a very diverse group of proteins contains alpha helix. The keratinins are a family of closely fibrous proteins, major component of tissues such as hair and skin. Keratin and myoglobin is globular flexible molecule whose structure is approximately 80% alpha helical and the rigidity is determined by the number of disulfide bonds between the constituent polypeptide chains. Hydrogen bonds in alpha helix is stabilized by extensive hydrogen bonding between the peptide bond, carbonyl oxygens and amide hydrogens that are part of the polypeptide backbone. The first and the last peptide bond component are linked to each other through hydrogen bonds. Hydrogen bonds are weak but they are collectively served to stabilize the helix. The amino acids per turn, each turn of an alpha helix contains 3.6 amino acids. The amino acid residues spaced 3 or 4 apart in the primary sequence are close together when folded in the alpha helix. Now, amino acids that disrupt the alpha helix, proline disrupts alpha helix because its amino group is not geometrically compatible with the right-handed spiral of the alpha helix. Charged amino acids like glutamate, aspartate, histidine, lysine, arginine, they can disrupt the alpha helix by forming ionic bonds or by electrostatically repelling each other. And amino acids with bulky side chains like tryptophan and large number of amino acids, example valine or histidine that branch at the beta carbon can also interfere with formation of the alpha helix. Now, if we talk about the beta sheet, it's the second form of regular secondary structure in proteins. They are less common than the alpha helix. The structure of beta sheet appear pleated. Beta strands are often visualized as broad arrows. Now, if we do the comparison of beta sheet and alpha helix, beta sheet are composed of two or more peptide chains. The hydrogen bonds in beta sheet are perpendicular to the polypeptide backbone, and parallel and antiparallel sheets. Beta sheet can be formed from two or more separate polypeptide chains that are arranged either antiparallel or parallel. The antiparallel beta-beta sheet polypeptide chains are arranged to each other with the N-terminal and C-terminal ends of the beta strands. Parallel beta-beta sheets polypeptide chains are arranged to each other with all the N-terminal of the beta strands together. And then the interchain bonds. The hydrogen bonds is formed between the polypeptide backbones of several polypeptide chains. And the interchain bonds, a beta sheet can also be formed by single polypeptide chain folding back on itself. In this case, the hydrogen bonds are interchain bonds. So these are parallel and these are then anti-parallel beta sheets. Now beta bands are reverse terms. Beta bands reverse the direction of polypeptide chain by helping it form a compact globular shape. They are usually found on the surface of protein molecules and often include charged residues. So beta bands is generally composed of four amino acids, amino acid that causes a kink and the amino acids with smallest R group. Now beta bands were given this name because they often connect successive strands of antiparallel beta bands. Beta bands are stabilized by the formation of hydrogen ionic bonds. Now non-repetitive secondary structure ends, loops and turns are best recognizable as forms that resemble neither an alpha helix nor a beta-braided sheet. These do not have a repeating element in their amino acid sequence. These structures are often found on the surface of the protein. Now, what is the supersecondary structural motif? Supersecondary structures are usually produced by packing side chains from adjacent secondary structural elements close to each other. Example, alpha helix and beta sheets are adjacent in the amino acid sequence are also usually adjacent in the final folded protein. Some common structural motifs combining alpha helix and beta sheets are beta alpha beta unit, Greek key, or beta meander, beta barrel. Now, what are motifs? A motif is an arrangement of small number of secondary structures replicating in many different proteins. A motif is a short recurrent segment of protein, three-dimensional structure that is found in a large number of different peptides. A motif may contain 2 to perhaps 7 or 8 elements of secondary structures. Example, the beta alpha beta motif consists of 3 secondary structures, a beta strand connected to an alpha helix connected to another beta strand. Motifs cannot exist on their own, they require the rest of the domain to maintain their conformation. Now, if we talk about tertiary structure of proteins, tertiary structure is the total 3D conformation of an entire polypeptide chain, including alpha helices, beta sheets, and any other loops, turns, and bends. The structure of globular proteins in aqueous solution is compact with high density of the atoms in the core of a molecule. Hydrophobic side chains are buried in the interior, whereas hydrophilic groups are found on the surface of the molecule. This eliminates the possibility that water molecule may become bound to these hydrophilic groups and disrupt the integrity of the protein. So the alpha helix and beta sheet structures provide maximal hydrogen bonding for peptide bond components within the interior of the polypeptide and involved in hydrogen bonds. Now, what is the structural domain? Domains are the fundamental functional and three-dimensional structural units of polypeptide. The polypeptide chain contains two or more domains. The core of a domain is built from combinations of motifs. Now, what are the interactions stabilizing tertiary structure? Disulfide bonds. A disulfide bond is a covalent linkage formed from the sulfhydryl group of each of these two cysteine residues to produce a cysteine residue. A disulfide bond contributes to stability of the three-dimensional shape of protein molecule and prevents them from denature in the external environment. Then we have hydrophobic interaction. Amino acids with nonpolar side chains tend to be located in the interior of the polypeptide molecule where they associate with other hydrophobic amino acids. In contrast, Amino acids with polar tend to be located on the surface in contact with the polar solvent. If we talk about hydrogen bonds, amino acid side chains containing oxygen or nitrogen bound hydrogen can form hydrogen bonds with electron rich atoms such as oxygen of a carbonyl or carboxyl group or peptide bond. Formation of hydrogen bonds enhances the solubility of the protein. Now, if you talk about ionic interaction, negatively charged groups such as the carboxyl group in the side chain can interact with positively charged groups such as the amino group in the side chain of polypeptide. Now, protein folding, which occurs within the cell in seconds to minutes, implies a shortcut to the maze of all folding possibilities. As the peptide folds, its amino acid side chains are attracted and repulsed according to chemical properties. The role of chaperones in protein folding. A specialized group of proteins called chaperones are required for the proper folding of many species of proteins, also known as heat shock proteins, interact with the polypeptide at various stages during the folding process. Some chaperones are important in keeping the protein unfolded until its synthesis is finished or act as catalysts by increasing the rates of the final stages in the folding process. It protects proteins as they fold so that their vulnerable exposed regions do not become tangled in unproductive encounters. Now, if we think about quaternary structure of proteins, quaternary structure is the arrangement of multiple folded protein or coiling protein molecules in a multi-subunit complex. So, protein denaturation results in the unfolding and disorganization of the protein secondary and tertiary structure, which are not accompanied by hydrolysis of peptide bonds. So, denaturation may be reversible into its original structure when denaturing agent is removed, and denatured protein are often precipitates from the solution. Now, denaturation agent can be heat, organic solvents, mechanical mixing, strong acids and base detergents, and ions of heavy metals such as lead and mercury. Now, what is protein misfolding? Protein folding is a complex trial and error process that can sometimes result in improperly folded molecules. So these misfolded proteins can usually tagged and degraded within the cell. Deposits of these misfolded proteins are restricted with a number of diseases, including amino acids. So, amino-acids build protein assemblies consisting of beta-pleated sheets. Accumulation of these spontaneously aggregating proteins called aminoids has been implicated in many degenerating diseases, particularly in the neurodegenerative disorder Alzheimer's disease. The dominant component of the aminoid plaque that accumulates in the Alzheimer's disease is alpha beta, and this peptide, when aggregated in a beta-pleated sheet configuration, is neurotoxic. So the alpha beta amyloid that is deposited in the brain in Alzheimer's disease is derived by proteolytic cleavages from the larger amyloid precursor protein. And the key component of these triangle fibers is the abnormal form of the tau protein. Now, if we talk about the prion disease, the prion protein PRP has been strongly implicated as a causative agent of transmissible spongiform encephalitis, including Creutzfeldt-Jakob disease in humans, staph in sheep, and bovine spongiform encephalopathy in cattle, popularly known as mad cow disease. So the prion protein tends to form insoluble aggregates of fibrils and is highly resistant to proteolytic degradation, similar to the amyloid found in some other diseases of the brain. Now, PRP is a host protein. The key to becoming infectious apparently lies in changes in the three-dimensional conformation of PRP. It has been observed that a number of alpha helices present in infectious PRP are replaced by beta sheets in the infectious form. In Alzheimer's disease, normal proteins after abnormal chemical processing take on a unique conformational state that leads to the formation of neurotoxic amyloid protein assemblies consisting of beta sheets. And in such cases, the infective agent is an altered version of a normal prion protein, that acts as a template for converting normal protein to the pathogenic conformation. So, what do proteins do? They provide structure, catalyze cellular reactions, carry out other tasks, and have multiple biological functions classified according to their biological roles. For example, there are enzymes that are a most varied and specialized group of proteins with catalytic activity. Right, they are all chemical reactions of organic biomolecules. The cells are catalyzed by enzymes. Then there are transport proteins. They bind and carry molecules or ions to organs in the bloodstream. Blood plasma. Lipoproteins in blood plasma carries lipids from the liver to other organs. Then we have nutrient and storage proteins. Seeds of many plants store nutrient proteins required for the growth of the germinating seedlings. Ovalbumin, the major protein of egg white, and casein, the major protein of milk, are examples of nutrient proteins. Then we have contractile or motile proteins. Some proteins endow cells and organisms with the ability to contract, to change shape, or to move about. Tubulin is the protein from which microtubules are built. We have structural proteins. Many proteins serve as supporting filaments, cables, or sheets to give biological structural strength or protection. Major component of tendons and cartilage is the fibrous protein collagen, which has very high tensile strength. Ligaments contain elastin, a structural protein. Then we have defense proteins, which defend organisms against invasion by other species or protects them. Immunoglobulin or antibodies are made by the lymphocytes of vertebrates and can recognize and precipitate or neutralize invading bacteria. So fibrinogen and thrombin are blood clotting proteins. Then we have regulatory proteins that help regulate cellular and physiological activity. Cellular response to many hormonal signals is often mediated by a class of GTP binding proteins called G proteins. Now, every amino acid has the same fundamental structure, which consists of a central carbon bonded to an amino group, a carboxyl group, and a hydrogen group. So there are 20 different types of amino acids in making proteins. Okay, the essential structure is the same in all 20 molecules. The molecule different in the side groups. So, as I said, the peptide bond is a covalent bond. They're polypeptides are polymers of amino acids linked together by peptide bonds with the amino group of one acid joining the carboxyl group of its neighbor. Amino acids. So, peptide bond is formed when the OH group from the carboxyl group of one of the amino acids is removed and hydrogen bond from the other amino acid is removed. So the OH and the H bond to form water. The formation of water from the two amino acids is called dehydration of synthesis. And the covalent linkage that is formed is known as peptide bond, and the molecule that is formed by the linking of amino acids is called polypeptide. Some proteins consist of a single polypeptide chain, and others called multi-subunit proteins have two or more. So individual polypeptide chains in a multi-subunit protein may be identical or different. If at least some are identical, the protein is sometimes called oligomers protein, as the units themselves are referred to as protomers. We've already talked about the different levels of structures. Right, we talked about protein peptide bond formation. Alpha carboxyl group of one amino acid with a side chain of R1 forms a covalent peptide bond with alpha amino group of another amino acid with a side chain of R2. By removal of molecular water, the result is dipeptide, two amino acids linked by one peptide bond. By the same way, the dipeptide can form a second peptide bond with a third amino acid with side chain R3 to give tripeptide. Repetition of this process generates polypeptide or protein of a specific amino acid sequence. Peptide bond formation. Each polypeptide chain starts on the left side by a free amino acid group of the first amino acid entered in the chain formation. It is termed N-terminus. Each polypeptide chain ends on the right side by free COH group of the last amino acid and termed C-terminus. So these are examples of peptides, dipeptides, tripeptides. Dipeptide can be aspartame, which acts as a sweetening agent used in replacement of cane sugar. It is composed of aspartic acid and phenylalanine. Tripeptides are GSH, which is formed from three amino acids: glutamic acid, cysteine, and glycine. It helps in protects against the free radical, which causes cell damage. Octapeptides, oligopeptide, polypeptide, proteins, etc. So, N-terminal residue can be identified by using a reagent that form bond covalently with its alpha NH group. Because the bond is stable to hot acid hydrolysis, the derivative of the N-terminal residue can be identified by chromatographic procedures after the protein has been hydrolyzed. Two reagents are commonly used. Sanger's reagent contains 1-fluoro-2,4-dinitrobenzene. It reacts with free NH2 group in an alkaline medium, and the compounds so formed can be isolated after protein hydrolysis and identified. Sanger was the first to sequence a polypeptide. He determined the complete primary structure of the hormone insulin. Then we have diacetyl chloride. The N-terminal NH2 group can also be combined with diacetyl chloride, 1-dimethylaminophthalein 5-sulfonyl chloride to form a fluorescent diacetyl derivative, which can then be isolated and identified. Then we have Edman degradation, a similar reaction with NH2 group and occur with the reagents with isocyanate, and thus became enables identification of the N-terminal amino acid. Edman and G Edmund and G. Baker have perfected an automated amino acid sequenator for carrying out sequential degradation of peptides where the phenyl acid has cyanate. Procedure. Automated amino acid sequences are now widely used with permanent very rapid determination of the amino acid sequences of polypeptides and permanent amino acid. Approximately so amino acids are determined sequentially from N-terminal end. So the phenyl thiohydrantoin amino acid liberated by is determined identified by HPLC. We have talked a lot about the different functions of proteins, catalyzed reactions, chemical signaling, storage, structural, protective, contractile, transport, etc. Talked about the different structures as well. The kinds of RNA, the class of RNA found in ribosomes is called RNA, ribosomal RNA. During polypeptide synthesis, rRNA provides the site where polypeptides is assembled. Then we have tRNA molecules, both transport the amino acids to the ribosome for use in the building of the polypeptides, and each position each amino is in the correct place on the elongated polypeptide chain. So human cells contain about 45 different kinds of tRNA molecules. Then we have messenger RNA molecules, just long strands of RNA that are transferred from DNA and that travel to the ribosomes to detect precisely which amino acids are assembled into polypeptides. So it follows the central dogma. The information passes from the DNA to an RNA copy of the gene, and the RNA copy detects the sequential assembly of a chain of amino acids. So genetic code, essential question of a gene expression is how does the order of nucleotides in a DNA molecule encode the information that specifies the order of amino acids in the polypeptide. So the experiment was so elegant that the result was so critical to understand the genetic code that we described in detail. Sukri and his colleagues reasoned that the genetic code most likely consisted of a series of blocks of information called codons. So they further hypothesized that the information within one codon was probably a sequence of three nucleotides specifying a particular amino acid. They added the number 3 because the two nucleotide would not yield enough combinations to code for the 20 different amino acids that commonly in the proteins. With 4 DNA molecules GTAC, only 42 or 16 different pairs of nucleotides could be found. However, these other same nucleotides can be arranged in 43 or 64 different combinations of 3 or more than enough to code for the 20 amino acids. So when they made a single deletion or two deletions near each other, the reading frame of the genetic message shifted and the downstream sick gene was transcribed as nonsense. However, when they made 3 deletions, the correct reading frame was restored and the sequences downstream were transcribed happily. So they obtained the same results when they made additions to the DNA consisting of 1, 2, or 3 nucleotides. However, the same nucleotides can be arranged in 343 or 64 different combinations, more than code for 20 amino acids. When they made a single deletion or two deletions near each other, reading frame of the genetic message is shifted and the downstream gene was transcribed as nonsense. However, when they made 3 deletions, the correct reading frame was restored and the sequences of downstreams were transcribed correctly. So they obtained the same result when they made additions to the DNA consisting of 1 or 2 of these nucleotides. So you see how the entry of one strand changes, how the entry of our strand uh, is normal. So the code is practically universal. For example, the code on AGA consists of amino acid, arginine, bacteria, humans, and all other organisms whose genetic code has been studied. So because the code is universal, genes transcribed from one organism can be translated in another, and the RNA is fully able to retain a functionally active protein. Similarly, genes can be transferred from one organism to another and successfully transcribed and translated in the new host. Many commercial products such as insulin used to treat diabetes are now manufactured by the placing human genes into bacteria, which often serve as tiny factories to turn out prodigious quantities of insulin. Genetic code. In 1979, investigators began to determine the complete genetic sequence of the mitochondrial genomes in question. Something was shocked when the investigators learned that genetic code used by the mammalian mitochondria was not quite the same as the universal code that has become familiar so far. In the stop in the mitochondrial genomes, what should have been a stop codon UGA was instead read as the amino acid tryptophan. AUA was read as methionine rather than isoleucine, and AGA read as AUG were read as stop rather than arginine. So thus it appears that the genetic code is not exactly universal. The first step of gene expression is the production of an RNA copy of the DNA sequence including the gene, a process called transcription. To understand the mechanism behind the transcription process, it is useful to focus first on RNA polymerase and remarkable enzymes responsible for carrying it out. RNA polymerase is best understood in bacteria. Bacterial RNA polymerase is very large and complex tech consisting of 5 units. So 2 alpha 5 subunits bind regular proteins, beta subunit binds DNA templates, beta subunit binds RNA nucleoside subunits, or sigma subunit recognizes promoter and initiates protein synthesis. Only one of the two strands of the DNA called the template strand is transcribed. The other strand is not transcribed and is called the not coding strand. The polymerase adds nucleotides to the drawing 3' end of the RNA chain. A eukaryote carry only one RNA polymerase enzyme, with eukaryotes have 3 different RNA polymerases ready. So this RNA is the tRNA. And transcription starts at RNA polymerase binding sites called promoters on the DNA template strand. A promoter is a short sequence that is not itself transcribed by the polymerase and binds into it. A promoter has differs widely in variety and efficiency. Strong promoters cause the frequent initiations of transcription as often as every 2 seconds in some bacteria. Weak promoters may transcript only once every 10 minutes. In bacteria, a severity of RNA is called sigma recognizes the one-nested sequence of the polymer promoter as Ine's RNA polymerase then. So importantly, this subunit can be detected in the -10 sequence without unwinding the DNA double helix. In eukaryotes, the 21-25 sequence plays a similar role in initiating transcription as it is the binding site for a key protein factor. Other eukaryotic factors then find one another, assembling a large question and to RNA polymerase begins to unwind the DNA helix. Unlike DNA's primary is now not required, the region containing the RNA polymerase is called uh, locally wound babble. Transcription bubble moves slowly down the DNA at a constant rate of nucleotides per second, leaving the room RNA stranded protruding from bubble. After the transcription and bubble passes, the now transcribed DNA is dim around as it leaves the bubble. Unlike DNA polymerase, RNA polymerase has no proofreading capability. In translation, that produces many more copying errors and application. Most genes are transcribed beautifully many times. So few faulty, harmful, that are harmful. At the end of the gene are stop codons that cause the formation of polydiacid bond to seize the RNA polymerase to release the DNA. And the DNA within the translation bubble to rewind. The simplest stop signal is a series of GC base pairs followed by a series of AT base pairs. So RNA transcript of this stop region forms a GC hairpin followed by 4 or more nucleotides. How does structure terminate transcription? The hairpin causes the RNA polymerase to pause immediately after the polymerase has synthesized it, placing the polymerase directly over the run of 4 cells. Fourier distance. The pairing of UB-DNA is the weakest of the 4 hybrid base pairs and is not quite strong enough to hold the hybrid strands together during the long pause. Instead, the RNA strand dissociates from the DNA within the transcription bubble and starts the transcription until it starts. A variety of protein factors and hairpin air loops are also dominating transcription of particular genes tonight. In translation, in prokaryotic translation begins when the initiator portion of an mRNA molecule binds to an RNA molecule in a ribosome. mRNA lies in the ribosome in such a way that only one of its codons is suspected at the polypeptide making site at any time. A tRNA molecule processing the complementary nucleotide sequence or announcing anticodon binds to the exposed codon on the RNA. Because this tRNA molecule carries a particular uh, amino acid and no other is added to the polypeptide in that position. As the mRNA molecule moves through the ribosome, successive codons of the mRNA are exposed in the series of tRNA molecules bind one another to the respective codons. Each of these tRNA molecules carries an attached amino acid which it adds to the end of the growing polypeptide. There are about 50-45 different kinds of RNAs available. Why are there 45 and not 64 tRNAs? How do particular new acids become associated with chromatically particular tRNA molecules? The key translation step with the pairs the three-dimensional sequences with appropriate amino acids is carried out by a remarkable set of enzymes called activity enzymes. So these are the different alphabets for the start and the stop signal. Initiation in eukaryotes and eukaryotes is difficult because they differ into different ways. The initial complex is far more complicated than in bacteria because 9 out of the protein problems may consist of several units. We talked about elongation and tRNA happy molecule with the appropriate anticodon copious proteins called algorithm. Proteins called elongation. In a passive transition along the RNA molecule, the movement reduces the initial tRNA to the E side and escapes from the ribosome whenever the grain related chain of the P side. So elongation of continues in this fashion until the chain terminating nonsense codon is exposed, like UAA, and nonsense codons do not bind to tRNA but they are recognized by the release factors. Proteins are released the highly newly made polypeptide protein from the ribosome. So this was back to the question that we started with. The answer will be RNA. If anybody has any doubt, they can raise their hand and unmute the voice and say, or I'll look into it at the end. So during protein synthesis in ribosomes, pellets uh, peptide bond formation is by RNA. Okay, that is the correct answer. So I'll give you 2 to 3 minutes. So, no doubt in this question, we'll move on to the next question for today. mRNA capping ensure a protection, translation, splicing, all of the above. So, I'll give you 2 to 3 minutes. If you know the answer to this question, you can unmute and let me know, and then we'll discuss. Okay, so the answer of this question will be all of the above. mRNA capping ensures protection, translation, splicing, all of these. Right? So when we need for the process of initiation, we need special promoter sequences at 10 and 35, right? So the sigma factor binds to the promoters. Helices fit into the major group. And in eukaryotes, several transcription factors assemble and bind to promoter. Additional sequences upstream CAT box, enhancers, silencers, etc. The regulation of transcription cis and trans regulators, cis factors, sequences upstream, Tata box, enhancers, etc. Trans factors are proteins that bind to promoter made by other faraway genes. Now there are chromatin structures also regulates transcription. Instrons prevent transcription factors from binding. In interphase, DNA is in 30 nanometer fiber phase bound around like nucleosomes. Now in eukaryotes, for elongation, the structure of RNA proto and in eukaryotes, special transcription sequence which cuts the enzyme cuts transcript and adds a long string of polyase. Now what is splicing? We have introns, like for example the human dystrophin gene has a huge intron. Splicing is critical sequences are in the introns, 5' GU, branch point A, 3' AG, all of these are in the introns. Now what is the detail of intron removal from pRNA molecule? If you see there is a cleavage at 5' exon intron junction and formation of a lariat structure. Okay. Branch sequence, then cleavage at 3' exon intron junction and ligation of the 2 exons. Branch junction structure involving an initial 2' 5' phosphodiester bond between C nucleotide from the 5' junction and the branch point sequence. Both the 5' splice side and the branch side are checked before any splicing reaction occurs, and the sequence must pass both identity checks. The U2 and U6 fold the pre-mRNA in a way that juxtaposes the 5' S's in the branch point, and cleavage at the 5' S's and addition of U5 bring the 3' and 5' splice sites together. So there may be special splicing enhancer sequences in the exons. Proteins and RNA bind to these sites and define the exons. Splicing errors can lead to diseases such as spinal muscle atrophy. Muscle splicing and introns. Group 1 introns in RNA able to catalyze their own splicing reactions. First example of an RNA enzyme. Why does it matter? Because it's important for ideas about the origin of life. Often called the RNA world. Polymerase 1 transcribes ribosomal RNA. Ribosome is used to make proteins. Has two subunits, large and small. Large subunit is composed of RNA 28S, 5.8S, and 5S and many proteins. Small subunit has 15S RNA and many proteins. In eukaryotes, 18S and 5.8S RNA and 28S RNA are adjacent with sequence repeating hundreds of times, transcribed as a unit and then cleaved. RNA genes and RNA production in E. coli. You see over here. All three makes tRNA and 5S rRNA. Transfer RNAs are necessary to make proteins that bring the correct amino acid. Each tRNA has a unique sequence with slightly different shape, order plus minus clover leaf. Transfer RNA anticodon at one end recognizes mRNA sequence, amino acid attachment site at the other, always CCA added after transcription. Anticodon at one end recognizes mRNA sequence, amino acid attachment site at the other, always CCA added after transcription. They often have several modified bases, uracil, ribothamidine. These modifications happen post-transcription. So, clover leaf structure of each alanine tRNA. Then we have polymerase C, promoted for 5S and tRNA are within the gene sequence or upstream. Okay, this is a model for intron removal by the spliceosome. Now what we do? We know about alternative splicing. Some genes have alternative splicing since the different exons are used in different tissues. RNA editing. In some cases, in some species, the final RNA molecule is much longer than the corresponding DNA sequence. Extra bases are added after transcription. Okay. Then we talk about gene expression. Eukaryotic gene expression. We have transcription, RNA splicing, capping, 5' poly A tails, and 3' regulation. Molecular control of transcription in eukaryotes. The transcription of eukaryotic genes is regulated by interactions between proteins and DNA sequences within or near the genes and sometimes from distance. Promoters of DNA sequences upstream of transcripts that initiate transcription. Promoter attacks RNA polymerase transcription start site. Enhances the simulate transcription. Silences inhibit transcription. Both are orientation dependent, position dependent, independent, can work at a distance from promoter and can bind to regulated transcription factors, position upstream or downstream sequences found in certain genes. The structure of a typically eukaryotic gene with its core promoter and proximal control region. The promoters take this and a polymerase comes and attaches to the promoter region. The pre-initiation complex is a large complex of proteins that is necessary for the transcription of protein coding genes in eukaryotes. PIC function which helps position RNA polymerase 2 over gene transcription sites and denatures the DNA. So what are the models for gene expression? Liver cell nucleus, brain cell nucleus, transcription of albumin gene at high level, transcription of albumin gene at very low basal level. Now what are activators? These regulatory proteins bind to DNA at distant sites known as enhancers. When DNA falls so that the enhancer is brought into proximity with the initiation complex, the activated proteins interact with the complex to increase the rate of transcription. These transcription factors transmit signals from the activated proteins to general factors, and these transcription factors position RNA polymerase at the start of a protein coding sequence and then release the polymerase to initiate transcription. Now, control of gene expression regulated by the control of transcription initiation, but gene expression can be controlled after transcription with mechanisms such as RNA editing or RNA splicing, mRNA stabilization and degradation, protein stabilization and degradation. RNA editing can be broadly defined as any site-specific alteration in RNA sequence during or after transcription, excluding changes due to processes such as RNA splicing. RNA editing is a process that changes the identity of RNA bases after it has been transcribed from a DNA sequence. Okay, so insertion or deletion during transcription nucleotide, post-translational nucleotide, conversion or substitution nucleotide editing such as editing AG, most prevalent in human, editing CU, editing UC. Now what are the unmatched messages? Swapping of out of a single base in a section of a messenger RNA alters the genetic information transcribed from the DNA before it is transcribed into a protein. Transcription is complementary strand of RNA is produced. 3 base sequence codes for a specific amino acid. Translation ribosome reads its codon. None of that. Different protein is produced. Now, RNA editing creates mature mRNA that are not truly encoded by the genome. For example, apolipoprotein B exists in two isoforms. Isoform 1 produced by editing the mRNA to create a stop codon and translated to create apolipoprotein B48 in the intestine tissue. Isoform 2 produced by the unedited mRNA to create apolipoprotein B100 in liver tissue. The post-transcriptional regulation introns are spliced out of PMRNAs to produce the mature mRNA that is translated, splicing, removing intron spliceosomes. RNA splicing recognizes different splice sites, including some exons in different tissue types, and the mature mRNAs in each tissue possesses different exons, resulting in different polypeptide products from the same gene. RNA splicing of transcripts makes it possible for a single gene to encode several polypeptides, and its mechanism generates protein diversity. So, post-transcriptional regulation or alternative splicing. The synthesis of an RNA transcript, the three stages of transcription are initiation, elongation, and termination. RNA polymerase binding and initiation of transcription. Promoters signal the transcriptional start point and usually extend several dozen nucleotide pairs upstream of the start point. Transcription factors mediate the binding of RNA polymerase and the initiation of transcription, and the completed assembly of the transcription factors in RNA polymerase to bound to a promoter is called transcription initiation complex. A promoter called a Tata box is crucial in forming the initiation complex in eukaryotes. As RNA polymerase moves along the DNA and twists the double helix 10 to 20 bases at a time, transcription progresses at a rate of 40 nucleotides per second in eukaryotes. A gene can be transcribed simultaneously by several RNA polymerases. Nucleotides are added to the 3' end of the growing RNA molecule. Now, if you talk about termination of transcription, the mechanisms of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA can be translated without further modification. In eukaryotes, RNA polymerase to transcribe the polyadenylation signal sequence, the RNA transcript is released. 10 to 35 nucleotides pass this polyadenylation sequence. And enzymes in the eukaryotic nucleus modify the RNA, mRNA, before the genetic messages are dispatched to the cytoplasm. So during RNA processing, both ends of the primary transcript are usually altered. Also, usually some intermediate parts of the molecule are cut out and the other parts are spliced together. So each end of the pre-mRNA molecule is modified in a particular way. The 5' end receives a modified nucleotide 5' cap. 3' end gets a poly A tail, and these modifications share several functions. They seem to facilitate the export of mRNA to cytoplasm, and the product protect mRNA from hydrolytic enzymes. They help ribosomes attach to the 5' end. But we talk about split genes and polyadenylation and RNA splicing. So most eukaryotic genes and their RNA transcripts have long non-coding stretches of nucleotides that lie between coding regions. These non-coding regions are called intervening sequences or introns, and the other regions are called exons because they eventually express species translated into amino acid sequences. RNA splicing removes introns and joins exons, creating an mRNA molecule with a continuous coding sequence. In some cases, RNA splicing is carried out by spliceosomes, which consist of a variety of proteins and several small nuclear ribonuclear proteins that recognize the slide size. What are the molecular components of translation? A cell translates an mRNA messenger protein with the help of a transfer RNA. The RNAs transfer amino acids to the growing polypeptide in a ribosome, and translation is a complex process in the terms of its biochemistry and mechanics. Molecules of tRNA are not identical. Each carries a specific amino acid on one end and it has an anticodon on the other end. The anticodon base pairs with the complementary codon on mRNA. So coming back to the question that we started with, mRNA capping ensures protection, translation, splicing, all of the above. The answer will be all of the above. Okay. Anybody has any doubt with this question? I will wait for 3-4 minutes. Please let me know. Okay, so if nobody has any doubt in this question, we can stop today's session here. Sorry, I got out. If nobody has any doubt in this question, we can stop today's session here and we'll meet again next Monday. Okay.